morning, everyone. How are you doing today? So I recognize that I am standing between us and a break, so just stay with me. I'll try and make it a little lively today so that I don't put you to sleep. I hope that you have enjoyed Rome so far. Hopefully you've gotten your fill of gelato and wine and really good Italian food. I know I've definitely been enjoying it. So my name is Cynthia Pacinen. I am the CEO of Sernova. Show of hands, how many of you have heard of Sernova before? And Modi, my BD colleague in the back, doesn't count. I see my friends at EvoTech, they also don't count because they know us. So not a lot of hands, maybe 6 or 7, I think, and that was including Victor from EvoTech and Modi, who's our Chief Business Officer. So I aim to change that today, and hopefully you'll go away understanding a little bit more about Sernova and how we aim to change the world. So we really do envision a future where chronic diseases are no longer insurmountable obstacles. So forward-looking statements, disclaimers, you're all familiar with these. So a little bit about Sernova. So we are a pre-revenue clinical stage biotech company. We are really focused on providing regenerative medicine focused solutions for chronic diseases using therapeutic cells, tissues, other molecules needed for that proper biological functioning. And so we administer these therapies to patients in a delivery vehicle that we're calling the cell pouch. And the cell pouch is not an overly sexy or fantastic name, so we are taking suggestions for what we could use as a name besides the cell pouch. So a little bit about our clinical program and then those assets that we do have in preclinical exploration as well. So we have one program in the clinic today, and that is focused on type 1 diabetes. Today we are using human donor islet cells, so that is cells from deceased donors. We also have 2 assets in preclinical development, and those include hemophilia A and postoperative thyroid disease. The slide that I'm showing you here now is probably my favorite slide in the entire deck. And just like the previous speaker at Innate Bio, it is really nice to hear what patients are actually saying about the therapy, right? Because then you get a good feeling for what is this like in reality. So this demonstrates the power of the solution that we at Sernova have in our hands. And I'm going to read the words to you because it's kind of a long quote. So the patient goes on to say, after completing the safety, tolerability, and efficacy study of Sernova's cell pouch for clinical islet transplantation, I can easily state how absolutely wonderful life is to be free of always thinking about how to manage my diabetes. After having T1D for 47 years and approximately 21,500 injections of various cow, pig, and synthetic insulins, 34,000 plus finger sticks, 1,500 urine tests, 15 years on the pump, low blood sugar reactions, blood tests, doctors, and more doctors' visits. I have now been free of the need for injectable insulin for 15 months. My only wish is that this could have been done sooner. So this quote was during COVID, so back in June of 2021. This patient is celebrating her 4-year anniversary of being insulin independent within a couple of days. So that means no exogenous insulin usage. When you think about what that means for a patient with type 1 diabetes who thinks every single moment of every single day about how to manage their condition, the impact it has on them, the impact it has on their families, and then also the broader ecosystem, our broader healthcare ecosystem. It truly is life-changing. We use that word a lot, the terms transformational, life-changing. In this case, though, however, it's really quite accurate. So a little bit about our pipeline today, insulin-dependent diabetes. We've got hemophilia A, postoperative thyroid disease. We're working with a number of different partners. I mentioned EvoTech a moment ago. We're also working with the University of Piedmonte in Italy on our hemophilia A program. We also have a collaboration with AstraZeneca on the research side of things, and we just started a new program with AstraZeneca, which is really exciting for us. When I think about where else might we grow as a company, I feel that we've only just begun to scratch the surface of how the cell pouch could be used for chronic disease applications. So we've done some work recently with LEK on doing a broad-based assessment of the landscape out there and considering, where else could Sernova go. And what we've come up with is by really narrowing down the funnel, is a group of probably 12 Tier 2 assets and then 4 Tier 1 assets that we aim to prosecute or move forward, of course, depending on funding, which we're actively working on a daily basis, which if any of you attended Tim's talk at the beginning of the year, or at the beginning of this session, you heard him mention how challenging the atmosphere still is out there. So something that we're focusing on every single day. Do a high-level summary on Sernova and kind of some quick takeaways for you to think about. So we have established what I consider to be a really strong proof of concept. So in our phase 1/2 study, it shows that a regenerative medicine-focused payload delivered in that cell pouch, it's capable of achieving clinically meaningful benefits. So this includes improved HbA1c levels. I'm going to show you a graph in a little bit. So this is HbA1c levels back down into the non-diabetic or pre-diabetic range. And then insulin independence. So in our first cohort of patients, which included 6, all 6 of those patients have seen HbA1c levels reduced back into the non-diabetic range. 5 of those 6 patients are insulin independent. So again, this means no exogenous insulin usage. We expect to be in the clinic in the second half of 2025 with our induced pluripotent stem cell islet-like clusters. This is work that we're doing in conjunction with our partner Evotec, which is headquartered in Germany. Planning for this study is underway. We've had some really positive meetings with regulators already. We've also achieved preclinical proof of concept for postoperative hypothyroidism and hemophilia A. We put out a press release back in, I think it was the November timeframe, and we were excited to share news that we had been granted orphan drug designation and rare pediatric disease designation for our Hem A work. So again, very important patient population. Both are being advanced and both are using an autologous approach, so different than our type 1 diabetes work, which is not an autologous approach and where we do need to use immunosuppressives. We'll talk about that in just a couple of minutes. We anticipate filing an IND for our postoperative hypothyroidism program by the end of this year, moving into the clinic in 2025. So we'll then have two programs in the clinic in 2025, which will be exciting. We're a small team. We've only got about 24 colleagues. So for us, that's quite a lot of activity. So let's talk a little bit about the cell pouch and what it is. I don't have a sample with me today, so I'll just describe it for you. It's about a credit card-sized device. It's about this, it's, again, size of a credit card, about a few millimeters thick. It's made of a few polypropylene mesh sheets of material. So very biocompatible, tried and true, been in a number of patients, millions of patients in other applications. So we recognize that it's a really safe material to put into the patient. What you're going to see on the left side of this slide is a pictorial of what the pouch would look like actually laying in the body of a patient. And once we place the cell pouch in the abdomen of the patient, we allow it to fully vascularize. This step is truly important because if we didn't allow vascularization to happen, you would see cell death. And we want to make certain that we provide as many therapeutic cells to the patients as possible. So in this vascularization or prevascularization process, what happens is that cells and tissues grow through those mesh sheets of material that have little pores in them. And so you think about the roots of a tree growing into the ground, and this is pretty much what happens in the patient's body. And so through this vascularization process, we are forming that really healthy environment so that when we put the cells into that pouch, after a couple of weeks of prevascularizing, they can go off and do their work. So place those cells in, the cells then begin to function, fulfilling the biological function that the patient was not able to do previously. And so what we have in this cell pouch with the therapeutic cells is a pseudo-organ of sorts, right? And in the case of type 1 diabetes, what we do after the cells have been in the body of the patient, transplanted into the body for a couple of weeks, we then begin to titrate the patient off of insulin, and they then become insulin independent and enjoy many, many clinically meaningful benefits. So a little bit about where we are today, where we aim to go tomorrow. So today we're on number 1 on the slide, so left-hand side. And we are using human donor cells, cadaver pancreas islet cells. We recognize that there are just not enough cadaver cells in this world to treat all of the type 1 diabetes patients. There are millions of patients around the world with type 1 diabetes. We aim to ensure that our reach is as far and as broad as possible. This is where approach number 2 comes in. So in parallel with running our current phase 1/2 study, we are well underway working with Evotec, our partners, on process development, additional preclinical studies, and then manufacturing work for our iPSC islet-like clusters. And we'll see some data in just a moment on those islet-like clusters. This will ensure our virtually endless supply of cells to treat patients. So this is the off-the-shelf model. We can provide them to centers around the world so that when patients present, they can be used immediately. A little bit about our phase 1/2 study that's ongoing. You can see in the middle of this slide, I call it a Superman slide because it kind of reminds me of the superhero Superman. And what you see in the middle of the body is where the cell pouches are placed in the abdominal region above the level of the muscle. So it's not an overly invasive procedure, comparatively speaking, relatively easy, 15 minutes or so to place the cells or the cell pouch into the body. It's a single-arm, multi-cohort study. Primary endpoints include safety and tolerability, as you would expect. The cell pouch is well tolerated. It does not fibrose. Patients don't feel the pouch in the body. Their movement, their activities are not impacted at all. Secondary endpoints are numerous, so we've got probably 30 endpoints. Key ones really include the survival of the islets in the pouch. So we do histological studies, so we are able to prove that those cells are living and thriving, which is very important to understand. We can see, interestingly, the 3 components of the islet cells, so we're able to discern the alpha, beta, and delta portions of those islet cells. Other secondary endpoints include HbA1c reductions into the non-diabetic range, circulating C-peptide, and then a reduction in hypoglycemic events, which we've been able to see in all of our cohort 1 patients. So we do have 2 cohorts. We are fully recruited for both cohorts, so a total of 13 patients. We're waiting on a time slot for our last patient. He had a 6-week cruise that was on his calendar, and so that transplant got pushed out just a little bit further than we had anticipated. What we've really done is to take this study as a learning opportunity, right? So between cohort 1, cohort 2, what did we learn? So we learned a lot about the dose and the density that is needed of the cells to achieve that clinical benefit, right? And so we moved from an 8-channel cell pouch to a 10-channel cell pouch. So for just a little bit more landscape in the body with a cell pouch, we get about 50% more capacity. And this is important because what we recognized in cohort 1 is that all of the patients flipped into insulin independence at about the same relative dose level. So islet equivalents per kilogram of body weight, all relatively the same, and so we needed more space. So that's why we've gone to a slightly larger cell pouch. We also have learned quite a bit about the immunosuppressive regimen. So cohort 1, tough immunosuppressive regimen. We used Tacro, MMF, etanercept. When we moved to cohort 2, what we did was to drop down the level of tacrolimus by about 75%. We have also varied the MMF, belatacept, etanercept, and what we're trying to do is to find that optimal cocktail or that regimen that we want to use going into our iPSC protocol. And our goal is to obviously minimize the protection to the islets while then also we're minimizing the effect to the patients while maximizing the protection to the islets. Okay, so a little bit about HbA1c. What you can see in the darker blue bars on this slide is those HbA1c values for patients before they were treated with the cell pouch and human donor islets. So you can see that the levels varied, and they weren't all in good control. So this was on what the patient and their clinician had defined as optimal insulin therapy. When they were treated with the cell pouch and the human donor islets, all of those levels came under nice control, and all were back down into that non-diabetic range. So really quite positive findings for the patient. So a little bit about the data and what that looks like for our iPSC islet-like clusters. So again, this is work that we're doing with our partners at Evotec. And the right side of the slide shows a direct comparison in diabetic mice of human donor islets. So that's the data that you can see in the black on both the top right-hand side and then the bottom right-hand side. And so the top right-hand side shows the circulating C-peptide levels. And what you can see comparing the blue to the black, they're virtually identical. And when you go down to the bottom right-hand side, you can also see in the oral glucose tolerance test, they are also virtually identical. So what does that tell us, right? So what that tells us is that Evotec has done a really good job at engineering and then producing those iPSC islet-like clusters in the exact right ratio of alpha, beta, and delta portions, right? And so we've been asked a lot, well, how do you know that using the human donor islet cells, when you move into the iPSC study, how do you know that you're gonna see the same thing? This is how we have good confidence, because we're seeing really good preclinical data. We've done studies in over 35 or 40 animals currently, and we're on task to do a number of other animal studies coming up across this year and into next year. And so finishing up, because I know that I'm over time right now, just a couple of developments over the course of the first quarter that we have not talked about. So one of the things that's really exciting for us is we now have proof of long-term islet survival in the cell pouch. So we had always assumed or guesstimated that the islet cells survive, right? Because otherwise the patient wouldn't be having those clinically meaningful and positive results. But now we have histological evidence that there is proof of abundant vascularized islets expressing insulin in the cell pouch for over a year. We believe this to be the first in the industry, and we're really excited to know that and to have definitive proof of that. We talked about the IND that we're going to be filing for postoperative hypothyroidism. We talked about our new research program with AstraZeneca. And then I'll finish up just telling you about a malignant cell study that was done. One of the most powerful aspects of the cell pouch for me, and I believe for others, is the ability to maintain, so contain within that cell pouch the therapeutic payload that is in the cell pouch. So what goes in that cell pouch stays in there, and you can fully retrieve the cell pouch in case there were any issues whatsoever. So one of our partners did a study with malignant cells. So they put malignant cells in the cell pouch, put it into some mice, waited for 90 days, took the cell pouch back out, no evidence of malignancy in the animals. And so this is really good proof that that therapeutic payload does stay within the cell pouch. And you think about what are regulators around the world going to want to see? They're going to want to see that whatever you put into the body of a patient from a cell therapy perspective, xenogenic cell therapy, gene-modified cell therapy, that it's contained and that it can be retrieved in case of any issues. Safety of patients is always paramount. So we are a small company. What you see in front of you is our high-level cap structure. So average daily volume, anywhere between 300,000, a little bit higher, a little bit lower. We have about 303 million shares outstanding. We're in the SMID, so small to lower mid-cap region at about 200 million market capitalization. And that is it for today. Thank you so much for listening. And if you have any questions, please feel free to chat with me or to chat with Modi, who is our Chief Business Officer. Modi's in the back, and he's waving his hand. So chat with either one of us or email us. We'd love to talk. Thank you so much, everyone, and enjoy Rome. Thank you.